Okay, welcome everyone. Good morning. When I said we would start at 9 a.m. sharp, that was Jewish Standard Time, and for that we're actually starting early. So, but we have a full morning with some really incredible things that I'm so excited to be sharing with you and to be learning about with you. So, so we want to get started on time. First of all, for those physicians who are trying to get CME credits, I apologize. I realized I made a mistake on the little sheets we passed out. There's one missing number. What you do there is don't call, but text. Text the number that's on the sheet, 323-531-539, and there should be one more 9, 5399. And that text has to come this morning. After the session's over, they won't register CME credits anymore. So make sure to text this morning, 323-531-5399. And I'll come by after if I'll answer your question. Okay, so this morning's program, first of all, the schedule for this morning. The schedule is, and I'll introduce him in just a moment, we are starting with Dr. James Kirkland, and I'll describe to you why we are in for such an incredible treat for this presentation in just a second. Then we have Rabbi Siegelman, who's also an incredible treat, who's also here already. He'll be following immediately after Dr. Kirkland. Then we'll have a very brief break, and then we'll go straight into, when I say break, I mean 5 minutes, but just enough time for to get more snacks or check your phones, whatever it is. Then we'll have Dr. Joyce So, our new Chief Genomics Officer, who is absolutely incredible. I'm very excited about that. And then we have our keynote speaker, Rav Asher Weiss Shlita, who has just landed. He's here, he's davening right now, and we'll be seeing him, Mirz Hashem, shortly. He will be our keynote. And we did write on the sheet that it was from 9 to 1. We do expect to finish, more like 12:30 in that ballpark. But just so that you should have your morning reserved. Again, I have the sheets for CMEs. I'll announce that a few times throughout. I also want to thank the Robert M. Barron Family Foundation, specifically Julie Barron-Platt, the director and Cedars-Sinai board member, for funding this. And so what is this? What are we doing? Before I introduce our speaker, so that you can understand. Hopefully, I don't know if it was totally clear what this is, because this is not just a typical medical ethics conference, not just a typical Yom Iyun or Morning of Learning. This is something totally unique. Basically, the story is that as rabbi of this hospital, I've been here now for just about 20 years. And, thank God people ask the rabbi a lot of questions. A lot of questions about the kosher kitchen. A lot of questions about the Shabbos elevator. A lot of questions about Kohanim, a lot of end-of-life questions, grief types of questions. But I noticed something happening. First of all, just working here and seeing, this is an academic medical center. And you might have noticed the construction across the street when you parked this morning. That is a 10-story research tower that's going up right now. The academic research investment in this hospital is profound. And as I've been getting to know the researchers and the labs and seeing the things that are being done there, it's absolutely incredible, mind-boggling things that some of them are a few years away from being at the bedside, some of them are still dreams, and some of them are starting to have impacts on patients today. And they're mind-blowing. In fact, frequently I get asked to share with people, what are some of the new innovations at Cedars-Sinai? And I started to think, why should I be the one, trying my best to share it? We should share this. What's happening? This doesn't have to be a secret. This is not intended to be private information just for the select few who happen to ask questions. And also, I notice this is one area that I'm not being asked questions. What about this research? Should we be advancing in these ways? How should we be advancing? And so I thought, okay, if they're not asking me, I'm going to ask myself, and I'm going to share. We need to talk about these things because Judaism, the Torah, has incredibly rich wisdom and guidance to offer. So my initial idea was a full day of learning, and we're going to bring all of our most incredible innovations and researchers and investigators and have them all present and with rabbis interspersed throughout. And then I just thought, I don't know if people are going to come for a whole day. And also, why do we need to do it once for a whole day? Why don't we start doing this on a regular basis? At given intervals and different styles. Sometimes we'll have debates. Sometimes we'll have, just programs for rabbis, just programs for doctors. Today is meant to be community, especially, people who are interested, but as broad as possible. and to really start asking these questions and to look at some of the things that are happening. So I figured we have a morning, and let me pick some of the most incredible and innovative talents. And one of the things that is getting a lot of hype, if you haven't heard about it, it is getting a lot of hype. Maybe, Google it, is, anti-aging, longevity. People want to live forever. And it's a hot topic right now. Whether you know it or not, it's a very hot topic. And there's a lot of hype. A lot of things that people think like, oh, I listened to this podcast, or I now understand everything that's happening, and people are buying all kinds of things, or, doing all kinds of interesting, somewhat questionable things to live forever. And I thought, we have someone here at Cedars-Sinai who's an actual expert, who's doing actual science on this. And he agreed to share with us. Dr. James Kirkland is an actual, real expert in aging and senescent cell clearances, as he will describe to us. If you don't know what that word means, don't worry, you'll understand soon. He studies cellular senescence. He discovered the first agents that selectively eliminate senescent cells. They call it Senolytics, if I pronounce that right. If not, he'll pronounce it correctly in a moment. He demonstrated that they delay, prevent, or alleviate multiple disorders and diseases. He published the first blood biomarker score indicating senescent cell abundance and the first clinical trials of senolytic drugs. He has published over 360 publications on the topic, and he is right now currently involved in over 120 interventional and observational clinical studies, 120 at the moment. He was the 2020 recipient of the Irving Wright Award of Distinction in Aging Research. Dr. Kirkland's laboratory here at Cedars-Sinai is internationally recognized as a leader in aging research and a world leader in the application of senolytic agents. His laboratory is also investigating the influence of spaceflight on cell senescence. You might be aware that Cedars-Sinai has been sending missions actually to outer space to study this, and his lab is at the forefront of that. They're creating fascinating, transformative clinics and programs that you want to know about. And so you get to hear it firsthand from the expert, from the source. Without further ado, Dr. James Kirkland.
Thank you very much. It's a great honor to be here. Our lab and myself moved to Cedars from Mayo Clinic a couple of years ago. We're very happy to be here. The research programs here, as Rabbi mentioned, are expanding incredibly, whereas in other parts of the country, as you might have heard, research is going downhill, especially basic biological research and clinical research. So it's a great honor to be here. So I'm going to talk about some recent advances and future directions in what we call geromedicine. That's the science of developing interventions that target fundamental aging processes. So we're always aging. We begin aging before conception even. So it turns out that Down syndrome, which is a disorder when the mother's egg cells age before they are even fertilized, results in accelerated fundamental aging processes, and clinical trials are about to begin of interventions that target fundamental aging processes for people with Down syndrome. So you're always aging. This is why the WHO got rid of the International Classification of Disease Code in ICD-8 called aging, because a 6-year-old is older than a 4-year-old, and a 6-year-old wants to be a 10-year-old. So we're always aging, so it's no longer a disease classification that physicians bill for. What is a disease classification now in ICD-11, which is just being discussed and implemented at the moment, is impaired intrinsic capacity, that is impaired capacity to respond to a stress, and I'll come back to that. So sometimes people, especially younger executives in their 20s and 30s, the billionaire types, great people, a lot of them, but they come and they ask, what can I do to live to be 200? And my question is, do you want to live to be 200 and feel like you're 200? The real question people should be asking is, how can I extend my health span? The period during life when people are living free of pain, cognitively intact, able to work if they want, able to move around, and feeling happy and well. So the notion behind geroscience is to develop interventions that will delay, prevent, alleviate, or treat the many, many conditions that are associated with operation of fundamental aging processes. And these can include diseases, disorders, and conditions in children right through to the elderly. So what we want to do is compress the period of decline towards the end of life. And arguably, it might be nice to live to be 95, be completely cognitively intact, continue working if you wanted, and just not wake up one morning, rather than living to be 120 and feeling like you're 120. So actually, we've got a problem around the world. So in looking at 182 World Health Organization member states, lifespan has increased quite dramatically over the last 100 years, largely because of treatment for TB, tuberculosis. So tuberculosis accounted for 25% of deaths until streptomycin and other agents came along in the middle of the last century. And childhood diseases and disorders were the leading cause of death. So and more recently, we've been able to do things like treating high blood pressure and so forth, which has resulted in an increase in the number of people who are older. But we have not had a parallel increase in health span. And if we look at the 182 World Health Organization member states, which sometimes the US is part of, sometimes not, over the last couple of years, the US is the worst for the difference between health span and lifespan. Especially women in the United States have a 13.7-year period during which health span has declined to the point where they've got 2 or more chronic conditions and are having problems. And these problems accelerate into something that we call multimorbidity. So people start getting one age-related condition, then they get the next, and then the next one comes soon after that, the next soon after that. And people wind up on one medication, then 5, then 10, then 15, and it keeps going up. So women in the United States have the worst gap between health span and lifespan in the world. But every country, including developing countries in sub-Saharan Africa and South America, are also experiencing this. The other thing that's occurring around the world is the percentage of the population that is elderly is increasing dramatically. The world population is actually decreasing. But the families are having less children than it takes to replace the population. And this is especially noticeable in East Asia, where in China there was a one-child policy for many years. And any of you who've been to China recently, and especially also Hong Kong, where the Hong Kong is the longest maximum, near maximum lifespan in the world, you see a lot of elderly people and not that many children. So fundamental aging processes, as I mentioned, operate throughout our life. They also operate across all the vertebrates, that's animals with a backbone, and many invertebrates, and some plants, and some single-celled organisms. So these processes tend to be conserved across evolution. So aging processes are very fundamental. And there are a number of things that in humans and other vertebrates we know can impact on the rate at which these fundamental aging processes occur and that can result in a decline in health span. I've listed some of them here. You're aware of a lot of them, like obesity, problems with diabetes, exposure to radiation, exposure to chemotherapy, exposure to trauma like war stress, PTSD. All of these things can accelerate the rate at which some of these processes operate, and there are other things that appear to be able to slow them down that occur in the environment. But there are a group of processes that we call the pillars of aging or hallmarks of aging. You can slice and dice them into anywhere from 4 to 15 categories, but they're all interlinked in what we call the unitary hypothesis. such that if one is altered, all the rest tend to be altered. And it doesn't necessarily matter which one came first. If you get an acceleration of one fundamental aging process, that drives all the others, and then they drive that first one, and you get a cycle occurring. I like to think very simply in terms of 4 processes. I'm going to focus on only one of them, but I could be talking about all of the rest during the remainder of this talk. So I'll talk about cellular senescence, and I'll get into some detail about what that is. Another group of processes involve inflammation of tissues associated with chronic fibrosis or stiffening of tissues. Another are all kinds of changes that occur in various molecules and cellular components, like changes that occur in DNA and proteins and sugars and lipids, in intracellular organelles like the nucleus of cells, the mitochondria, endoplasmic reticulum, Golgi, and other cellular components. And then another group of processes involve stem and progenitor cell dysfunction. So you can form new tissues, it turns out, throughout life. We once thought you had a fixed number of heart cells, for example. You can form new ones, even in very old age, with some of the interventions that are being developed. But what happens with increasing age is that progenitor cells, or so-called stem cells that can give rise to new tissues, their capacity to do so alters. And they tend to alter in such a way that bad guy progenitor cells, for example, the ones that give rise to cells that eat up bone, increase, where good guy progenitor cells, for example, the ones that make new bone, osteoblastic progenitors, decrease. So there are changes in both directions in progenitors, but generally these fundamental aging processes favor the bad guy progenitors winning out over the good guy ones. And then the effects of these fundamental aging processes are very wide. So aging is the biggest risk factor for virtually any condition you can think of. If you look at the risk of having a heart attack or stroke, it's increased 2 to 4-fold by having a positive family history, a high blood pressure, a high blood sugar, not getting enough exercise, smoking, and so forth. But if you're 80 compared to 30, your relative risk of having these conditions is increased by 1,000-fold. So if you draw a histogram, a bar graph, and you put aging as a risk factor for virtually any condition you can think of, you don't even see the other conditions. So the things that we commonly think about that can be accelerated by these processes include multiple diseases and disorders, both acute and chronic. People always think in terms of chronic diseases, but COVID can be much worse in an elderly person or a person who's had chemotherapy and radiation or a person with obesity, partly because of these fundamental aging processes and partly because of the senescent cells that develop. Recovery from surgery can be delayed and so forth. And there's decrease in what we call resilience. So that's incomplete recovery after infection, surgery, wounding, or chemotherapy. There can be decreased reproduction occurs progressively. Development of aging phenotypes. You know, when you look at a person, you can tell how old they are roughly by how they appear. And decreasing remaining survival, as well as the geriatric syndromes that can occur even in younger people who, say, have been exposed to intense doses of chemotherapy and radiation. Things like cognitive impairment, frailty, sarcopenia, that's muscle loss, falling, immobility, and so forth. So I'll talk about cellular senescence briefly as a paradigm fundamental aging process. So cellular senescence, any kind of cell in your body can become senescent, whether it's dividing or not. And senescence is what we call a cell fate. It's like a cell dividing or a cell differentiating into a specialized cell type or a cell undergoing programmed cell death or apoptosis. It's a cell fate. It can occur to any kind of cell, as I mentioned, at any point during life, and it occurs across the vertebrates, many insects and invertebrates, some plants, even some single-celled organisms. And what senescence essentially entails is essentially irreversible loss of ability to divide, but also resistance to dying. So these cells can stop dividing, but they persist. Senescent cells are normally only removed by the immune system. They don't die on their own. So I'm not going to go into detail about this, but senescence can occur, cellular senescence can occur in response to stresses. So a cell can make a fate decision. Do I die? Do I ignore the stresses around me, or do I undergo a state of senescence? And the kinds of things broadly that can drive senescence are things like replicative stresses, that is, dividing a lot, or detection of outside damage signals, or inside the cell or outside the cell. Senescence is a cell fate that's necessary. If you stop senescence from occurring, you will get cancer because it's a defense against cancer. So a cell that divides repeatedly and has DNA mutations and oncogene expression can enter a state of senescence whereupon it stops dividing, which protects you from the cancer developing, and then that cell is removed by the immune system if the immune system is working properly. But unfortunately, senescent cells express immune evasion signals and can trick the immune system into believing that they're not there. Senescence is important during development for reshaping of organs. The first organ to disappear as we develop is something called the mesonephros. That's the kidney in the fetus that produces urine into the mother's fluid around the baby. And it has to be gotten rid of so the regular kidney can start working just before birth that delivers urine to the bladder and outside. And that occurs through a process of senescence. Later organs like the ovaries and the thymus and so forth involute because of senescence in later life. Senescence is important for delivery. So senescent cells form in the placenta, and during the last 5 days of pregnancy, they produce the factors that drive the baby through the birth canal. So it's necessary to have senescence occurring. In fact, some species like elephants, they're of course much bigger than we are. They've had their cells divide multiple times. They live longer than we do, but they rarely die of cancer. Very few cases of elephants dying of cancer. And this is partly because they form senescent cells very rapidly in response to cancerous mutations. But what we don't understand is how their immune system gets rid of those senescent cells. And the same with Wright's whales. They're much bigger than we are, but they don't die of cancer. So senescence is necessary. You don't want to interfere with the process occurring. But if a senescent cell develops, you have to be able to get rid of it. Your immune system has to be able to get rid of it. The faster, the better. Because over time, these senescent cells start developing more and more mutations, and through what we call transpositional events. Sometimes that's internal things called line 1 elements, sometimes viruses like HIV virus or human papillomavirus can act as transpositional elements within senescent cells. Over 2 to 4 months, they can start having a lot of mutations. They can escape senescence, whereupon they appear as cancers. So there's a belief now, an increasing belief, there were 3 Cold Spring Harbor conferences on looking at senescence and cancer, that so-called cancer stem cells or cancer progenitor cells or quiescent cancer cells are actually senescent cells. So you want to be able to get rid of senescent cells once they're formed. Furthermore, senescent cells develop what we call senescence-associated secretory phenotype, or SASP. And this can involve production of a lot of proteins, peptides, various reactive molecules, non-coding nucleotides, that's various forms of DNA and RNA that can get produced by these cells, and a whole lot of other things that can cause damage to other cells. They can form bridges to other cells with little channels and inject things into those cells around them. So senescence can spread. Not every senescent cell is tissue damaging. About 50% are, and 50% aren't. So we'll get to the difference between the bad guy and what we call, not necessarily good guy, but ticking time bomb senescent cells that are silent and not damaging tissues around them. Senescent cells defend themselves from being killed by what we call SCAP, senescent cell anti-apoptotic pathways. And we figured that out. I'll go to this first. If you transplant small numbers of senescent cells that are in a mouse, and you make the senescent cells from that mouse's own ear by taking a clip of the ear and then exposing those cells to radiation or chemotherapy, And then you take the mouse's own cells that have been made senescent and inject them into the abdominal cavity. If you inject above a certain number of senescent cells, 1 million senescent cells into a middle-aged mouse, that equals 1 in 10,000 cells in the transplanted mouse is senescent. That's sufficient after a few months to make the mouse become frail. And it is trouble running on a treadmill, trouble with hanging endurance, trouble with grip strength. And then after a lag period, these mice that are transplanted with senescent cells versus mice that are transplanted with in the same way but with non-senescent cells that weren't exposed to radiation or chemotherapy, the ones transplanted with senescent cells die earlier. And they die earlier of all of 250 age-related diseases in mice that we looked at that mice naturally die from later in life. So cellular senescence drives a whole lot of things, like basically everything out of these 250 conditions that will kill a mouse. Conversely, if we get rid of senescent cells genetically, and in this case we used a genetic trick to get rid of what we call highly P21 expressing senescent cells, they're more important than the P16 ones that you might have heard about in this regard. We can delay onset of age-related diseases and disorders as a group. So it looks like we've met what we call cost postulates for causation. Senescent cells are associated with many age-related conditions. If you put them in, you make the conditions, you accelerate them. If you get rid of them, you decelerate them. So this is the way that Koch showed that bacteria cause infections. So we used a hypothesis-driven approach. This was quite a number of years ago. We started working in 2004 on this, but we finally figured out how to do this in around 2013. We found that, as I mentioned, around half of senescent cells are bad guy senescent cells, and they're killing cells around them, much like some cancer cells, like lymphoma and certain kinds of leukemia cells, kill cells around them, yet they themselves don't die. So that led us to ask, how do they defend themselves from dying, despite the fact they're killing cells around them? Why don't they commit suicide with the factors that they themselves are producing? And we used early bioinformatics approaches and proteomic and sequencing data and worked with Jonathan Wren at University of Oklahoma, who had 7 Cray computers in a row. He's the person who invented text mining back in the 90s. And we found that there's a network of pathways that senescent cells use to defend themselves against the things that they're producing to kill cells around them. And then we used what we call RNA interference approaches to ask, if we target some of these pathways in the network, can we allow senescent cells to die? So we looked at human fat cell progenitors, which are probably the largest group of progenitors we have in humans. And we looked at human endothelial cells. And we found that different kinds of human cells depend on different elements within this network to survive. So different kinds of senescent cells use different SCAPs or pathways within the network to prevent themselves from dying. We then went to the Broad Institute in Boston. We were working in Boston at the time, and found that and looked at for things which would target the same pathways that we'd identified and confirmed with the RNA interference approaches. And we came up with a list of drugs and natural products that should arguably kill certain kinds of senescent cells. We found 40 of them. They all turned out to be what we call senolytic. We decided to focus early on agents that could be given orally and have a safety profile, that could be given in humans, and that have what we call a short elimination half-life, because it takes 2 to 6 weeks for new senescent cells to form. And as I mentioned before, they don't divide. If they do divide, they divide as cancer cells, but they typically don't divide. So we decided we wanted to use what we call a hit-and-run approach, intermittent administration of these agents. They'd be there for a very brief amount of time, because we found that it only takes 30 minutes or so of exposure to a senolytic to initiate irreversible cell death in a senescent cell, which takes about 18 hours to complete. So using that original mechanism-based approach, we found a lot of senolytics, and others did as well. And now there are other ways of finding senolytics using high-throughput screens and then looking at human populations to see are there particular drugs that are associated with delays in onset of age-related diseases. And are those drugs, in fact, acting in part on fundamental aging processes, including senescence? Two of them that came up were zoledronate that we've been working on with the group at Oxford for a number of years. We've had a couple of papers out and another coming on it. It turns out to be a senolytic drug. It's given once a year, once every 6 months for treating osteoporosis, and SGLT2 inhibitors, which are used for treating diabetes. I'll come back to that. We've discovered now, while at Cedars, immunomodulators that will do this. One of the people in our group, Masayoshi Suda, when he was in Japan before he moved to Mayo and then to Cedars with me, discovered the first vaccine against senescent cells. And then Miriam Tilton, who was at Mayo at the same time as we were, but moved to University of Texas at Austin, discovered that these cells are susceptible to mechanical influences. And then Baram Abu Daya, who moved at the same time as I did from Mayo to Cedars, he's in gastroenterology here, he discovered that electrical fields of the right type can kill senescent cells. And in fact, he's got a phase 2 clinical trial using high voltage, low amperage electrical impulses to kill senescent cells in the small bowel. And he's beginning to find that that's helping to treat diabetes. So another drug that we found is senolytic, I won't go into it, is fisetin. It's closely related to dasatinib and quercetin, which are the two that I showed in that earlier slide about the fat cell progenitors and endothelial cells. I'm just mentioning those three compounds because they're the main ones in clinical trials at the moment. And as I mentioned before, it just takes a brief exposure to these agents to kill senescent cells. If you take fat tissue explants from obese younger individuals, which are full of senescent cells, and compare them to lean younger individuals, it just takes a very brief exposure. We found that these senolytic drugs, if we radiate a single leg on a mouse with enough radiation that after 2 to 4 months, the hair on its leg goes gray and it has trouble running on a treadmill, a single administration of these drugs can restore its ability to run on a treadmill to that of wild-type mice for the next 7 months. So treating with senolytics is much like treating with antibiotics. You're getting rid of a cell type that, and once it's gone, it's gone. So this is like treating a urinary tract infection, where often nowadays we sometimes use a single dose of several antibiotics to stop the urinary tract infection. So this is different from the kind of thing that you think about with high blood pressure, where you're giving a drug every day all the time and trying to achieve a constant level to maintain blood pressure. Here we're getting rid of a bad guy cell type, much like getting rid of bacteria or viruses. We found that in old mice we could alleviate physical dysfunction. We found that if we transplant cells from obese individuals, humans, into mildly immunodeficient mice that can accept these implants, the mice that are transplanted with adipose tissue from obese, diabetic, younger human subjects that have senescent cells in them, compared to non-obese individuals, which don't have many senescent cells, we found that their glucose tolerance and other measures of, especially their insulin tolerance, is decreased, and that senolytics would reverse that if we gave that to the mouse that had the human transplant in it. We work with Stefan Tullius a lot in Harvard to look at heart transplants, and this is one thing we want to do at Cedars because we've got the biggest heart transplant program in the world. We want to move this into human trials when we can. But we found that transplanting hearts from old mice to young mice, the hearts from the old mice contain senescent cells. Senescence spreads from those hearts to the young mouse's own tissues, not because the senescent cells move around, but because of the factors they're producing as part of their secretory phenotype. We found that if we treat the donor mice, the hearts themselves, or the recipients with senolytics, we could improve survival after heart transplantation. I mentioned these SGLT2 inhibitors, which are commonly used for treating diabetes. They don't kill senescent cells directly. What they do is they enable the immune system to get rid of them. We don't understand quite how. But again, it was Masayoshi Suda who was part of this when he was still in Japan, working with Toru Minamino, and he's now at Cedars, who discovered this. I mentioned before that senescent cells, especially the bad guy ones, can hide from the immune system, much like cancer cells do. So you might have heard about these PD-L1 agents, which are commonly used. They're the leading treatment now for many cancers. Senescent cells use different immune evasion than cancers do. They use PD-L2 and certain other things. And we found while we're at Cedars, and by way of disclosure, Cedars has some patents on this. I'm not allowed to make money out of it, but Cedars might. So from a disclosure point of view, that's one disclosure to make. But these agents appear to allow the immune system to clear senescent cells. And we worked with others. Salim Chaib now, who is first author on this, is an assistant professor at Cedars. He moved with us from Mayo. We discovered another group of drugs, again, while we've been at Cedars, and again, by way of disclosure, Cedars and Mayo share a patent on this. Again, I don't get anything on purpose, because I'm doing clinical trials, so I can't. I don't even want to, But these drugs, what they do, I mentioned that 50% of senescent cells don't produce factors that kill cells around them, and therefore are resistant to senolytics. These cells, though, we found with infections, for example, coronavirus, can be rapidly converted into bad guy senescent cells within 3 hours, and they have 1,000 to 10,000-fold upregulation of production of factors that are harmful within 3 hours. So they're sitting there quietly. You don't know that you've got them. Then you get an infection, and you get very sick. So we capitalized on that, and we developed drugs that act the same way that these viruses do. In the case of coronavirus, it's an RNA virus, and it acts through something called TLR3. We developed drugs that would activate this, and we found that we're able to clear more senescent cells by giving senolytics, and then senescentizers, and then another round of senolytics. So we're using this for cancer treatment. We're hoping to use this for cancer treatment, because radiation or chemotherapy can make cancer cells either die or enter a state of senescence. They can escape that state of senescence and come back as more dangerous cancer cells that are chemo and radiation resistant. You've all heard of this and probably known people who've had a cancer come back, and then they're told, well, we can't give you chemotherapy or radiation anymore because it's resistant. So this is a way of arguably, hopefully, getting at the cancer-harboring senescent senolytic resistant cells that are sitting there mutating and waiting to escape as a chemo and radiation-resistant cancer. So in preliminary studies, this shows triple-negative breast cancer, that we might be able to do something about this. And we're working, people are probably in the lab at the moment working on this. We view this as a very high priority. So there's emerging evidence in preclinical models for benefits on a whole range of conditions that I don't have time to go into, including, as the rabbi mentioned, space travel and space radiation. And one of the things that I've been privileged to be able to be principal investigator on is something called the National Institutes of Health Translational Geroscience Network. So it's a network of 8 institutions that are full members and a whole lot that are affiliated. And what we're doing is helping people design clinical trials, not just of senolytics, but of other gerotherapeutics, things like NAD precursors, sirtuin agonists, metformin, rapamycin. Things like metformin and rapamycin, incidentally, act by inhibiting the secretory state of senescent cells. Senolytics result in an increase in NAD because they kill off an enzyme on cells called macrophages that degrade NAD. So all these processes are highly interlinked. And we're helping people with the Food and Drug Administration and with making sure consent forms are common across trials, making sure that if subjects agree, that we can keep their samples indefinitely and share them amongst groups and do reverse translation. That is, be able to take materials from a clinical trial, say blood or urine or tissue biopsies or data from a subject, and then take those things back to the basic science laboratory to inform the next round of experiments. We call that reverse translation. So we've, Cedars Ethics Committee and the IRB have been working on how do we design these forms, and we've come to consensus about that. We have common forms now. We're also developing forms for what we call N of 1 trials. That is, if people insist on taking things and we tell them not to because there's no evidence for it, will they give us samples before and after they do it? We tell them don't do it, sign a consent saying don't take this thing because we don't know yet if it works or if it's harmful. But if you're insisting on taking it, let us get your blood, your urine, test you before and after, and do that through a few cycles. And at least we can tell you if you're harming yourself, which most of the time they probably would be. But also we can inform the broader scientific community and aggregate these data. We have something called Facility for Geroscience Analysis, which moved from Mayo to Cedars now. So we've got a large laboratory in Pacific Design, and eventually we might be in the new research building, where we're measuring all kinds of things from across these trials, from around the United States and around the world. Many of the trials are occurring in Northern Europe, some in the Middle East. Canada's set up a parallel network, and Britain is in the process of setting up one. And then we biobank the samples, and we have shared data analysis, which is done at Wake Forest University by Steve Kraszewski and his group. So currently, we've got around 120 clinical trials underway, some of which are completed. And they're for conditions right from childhood conditions to things like frailty or Alzheimer's disease and various cancers in the elderly. Our strategy, because we don't know if these things work and people should not be taking these things until clinical trials are done, is to go after serious conditions for which there are no good current treatments or where treatments are incomplete. Because we don't know anything that sounds too good to be true is. We've got to know the downsides of these things. So we're starting off with serious conditions. We're not going for prevention in healthy 30-year-olds. And not only that, if people don't have senescent cells, why would you remove senescence? Why would you give them a drug to remove senescent cells? So several clinical trials have come out. The first two were in 2019, and one of them looked at idiopathic pulmonary fibrosis, which is a fatal lung disease associated with senescence. And it was a phase 1 trial, open label, a lot of problems with that kind of trial, but the results look promising enough that later trials are going on, including improvements in decreases in frailty, which is the main thing that these people get, especially in short physical performance battery. Another early trial in 2019 showed that we could clear senescent cells from humans, younger women with obesity, diabetes, and early renal failure. And we did biopsies on them before and after a brief course of these agents, and we found that they were actually removing senescent cells from humans. These fundamental aging processes tend to congregate within structures in tissue. So where you find senescent cells, you find fibrosis occurring, you find inflammation occurring, you find stem and progenitor cell dysfunction occurring, you find low NAD, you find increased mTOR, the target of rapamycin. They tend to occur in foci in tissues. You know about it in the brain, where you see plaques and tangles in Alzheimer's disease. In fat tissue, there are crown-like structures where these things are localized. In other tissues, you find little spots where these processes operate. I don't have too much time to go through it, but I mentioned we're looking at biomarkers, and we're trying to come up across the trials with biomarker scores. This is an early score looking at, that gives us an idea about senescent cell burden in people, and we found that Senolytics decrease it. These are just some of the analytes that we're looking at Cedars across all the trials from around the world. We're getting samples coming in from all over the place. Some things are good in youth that go down with age. Alpha Clotho is one of them. Alpha Clotho was the Greek goddess of fate. She holds the spool of life in her hand. She's the one in some cemeteries that looks down on the dead. Alpha Clotho goes down with aging in mice and in humans. It goes down in Alzheimer's disease. It's produced in the brain, the liver, the brain, the kidney, and to some extent fat tissue. If you overexpress it in mice, they live a lot longer and are smarter than other mice. Turns out that Senolytics make it go up in 20 out of 20 people that we looked at. So what we're trying to do is take the good guy and the bad guy biomarkers, come up with composite scores, and in each trial, cherry pick in one trial, and then test prospectively in the next one whether composite scores predict the kinds of results that we see and go along with clinical improvement. We're not interested in biological clocks. We're interested in things that change with interventions and predict clinical improvement. So far, we've found safety with some of these agents. Doesn't mean it'll continue. An early stage trial was done with Beth Israel and Heba Rehab in Boston. I've got very close collaborators there, Lou Lipsitz and Doug Keel and others. And we found a very, very early trial in Alzheimer's disease. We actually found improvement in the Montreal Cognitive Assessment Score. Similarly with osteoporosis, we did a trial with Sandeep Khosla at Mayo, and we found some improvements. And it's particularly the people in both these trials who have the most senescent cells who benefit the most from senolytics with these diseases, which you'd expect. We're working with Stefan Tullius at Harvard on trying to rehabilitate organs from older individuals so they can be put into younger people. So we're throwing away 24,000 kidneys a year because they're from older donors in the US. They're just being thrown away. That would, if we could use those kidneys, that would solve the entire organ shortage. With pancreases, we're throwing away pancreases from anybody over 45 if we're transplanting a whole pancreas. So we figured out that on LifePort machines and other machines where there's warm or cold perfusion, where the organ is being transplanted from the brain dead or dead donor to the recipient, we can add these drugs to the wash, which is in those perfusion systems, and get rid of senescent cells. So we're hoping that we can get to the point of being able to reuse organs from older people and younger individuals that are currently being thrown away. We're doing veterinary trials. We've got a positive trial now with improving wool production in old sheep, quality, color, hair thickness. And these veterinary trials look interesting. That's using a shampoo. And there are all kinds of other unusual trials and new trial designs coming along. So in conclusion, it appears that persistent senescent cells, it's the persisting ones that are damaging, can contribute to inflammation, fibrosis, stem and progenitor cell dysfunction, spread senescence both locally and at a great distance, and multiple disease and age-related disorders. The target of senolytics is senescent cells, not a single molecule or pathway. So we often combine them, and like with antibiotics, we don't necessarily care what the molecular target is. We care about the cellular target, senescent cells. We were using penicillin for 30 years before we figured out how penicillin worked. It was killing bacteria, so we used it. So fortunately, it's the branch of the FDA that deals with infectious disease that's dealing with us, because they understand this. Other branches of the FDA don't. Senolytics appear to attenuate inflammation, fibrosis, and so forth in animals and cell culture and other preclinical systems. A hit-and-run approach rather than continuous administration appears to be most effective, and that's how they're being used in the clinical trials that are planned and underway. They appear to delay or alleviate multiple diseases and improve tissue regeneration. We got new heart cells forming, for example, in very old mice, new brain cells forming, and enhance health span in mice, but we don't know if they work in humans. And as the rabbi mentioned, there's a lot of hype about some of these things. We don't use the word anti-aging. I mean, why? A 4-year-old wants to be a 6-year-old. Don't use the word anti-aging. And we'd kill all our cells. We're all aging at the moment. Why would we do something that's anti-aging? And we don't talk about lifespan at all costs. We're interested in, first of all, going after, being a physician, serious conditions that are life-threatening and that are causing a lot of pain and disability. Next, we'll go after secondary prevention and people who are at high risk of those kinds of things. But it'll be a long time before we get to thinking about these things in general populations. You know, so we're starting as physicians with the very ill and asking if we can help. And especially, how do we sequence these agents? Do we give these things with disease-specific interventions? Will there be synergy? It's looking like there could be for fibrotic diseases and other conditions. How do we go after various cancers where we've made cancer cells senescent by chemotherapy and radiation? But we don't know yet. These things are not things people should be buying from Amazon. We looked at some of the things that are sold by Amazon that are branded as Senolytic, and we did mass spectroscopy on 25 different products that claim to be Senolytic, and we found a lot of them contain milk powder because they're not regulated. And the amount of material in the capsules was highly variable, and there were toxic byproducts and things. So we have to be very, very careful about this. Lifestyle interventions is an even bigger black box. We don't know if exercise really works and what kind of exercise at this point. We don't know about diets. You hear all kinds of stuff about that. We're not certain. There are, of course, medical reasons for going on particular diets, like if there's high LDL cholesterol, LDL calculated cholesterol, there are particular diets that work. But with fundamental aging processes, we don't really know yet. Sleep looks like it's fairly positive. That one I'd go for, especially getting rid of sleep apnea. Stress is another one that's going to be very important. I mentioned that PTSD and anxiety can drive senescence. Various military in certain parts of the world, especially the Middle East, are very interested in this because PTSD appears to be transmissible from mothers who've experienced PTSD to their children conceived 10 years after their exposure to PTSD, when we find acceleration of fundamental aging processes in their children. So this can skip generations. So we really have to do something about war and, stopping this kind of thing from occurring. But again, we have to study these things. So the most important thing is to study them. And I'll just end by saying there's a huge number of people involved in this. I'm only the spokesperson for many, many, many people. And we're very grateful to the funding agencies that have supported this work, and very grateful to be at Cedars, where leadership here really understands, more than almost anywhere I've been, the potential promise of these and other new medical approaches. And where Cedars is ramping up research, where other places are ramping research down and being worried about high patient throughput and making profits, Cedars is investing, if they make profits, in the poor on the streets and in research. And I think I just feel totally aligned with that. Thanks.